Welcome to the University of Michigan Dentistry Podcast Series, promoting oral health care worldwide.
This segment is to review lymphatic tissue and the organs associated with the lymph system. There are the lymph nodes, there's lymph nodules, which are aggregations of diffuse lymphatic tissue, the spleen, and the thymus. Now, also included in this system is the tonsils, and we won't discuss them on this segment. First, I want to talk about the lymph node. The lymph node is surrounded by a capsule. It's very important to recognize this. This distinguishes it from the diffuse lymphatic tissue or the lymph nodules. Now we're going to go to the microscope and look at a lymph node. At the center of the node is the hilum, and this is connective tissue stroma. This happens to be where the blood vessels enter and exit from the lymph node and the lymphatics exit. This is connective tissue and vessels, both blood vessels and lymphatics, exiting and entering the node. Now let's look out in the periphery, and you see there's a different kind of tissue out here. This lymph node then has a medulla. The medulla is here, and a cortex on the outside. Now notice the capsule, the connective tissue capsule, on the periphery of this node. The node is a lima bean-shaped object surrounded by a connective tissue capsule. It has a cortex with these thousands and thousands and millions of small cells, cells of the lymphatic system, and then we have the medulla here. Now at medium power, let's again look at this lymph node. This is on the periphery of the node, the capsule to the left, the connective tissue capsule, and medullary tissue. Notice the thousands and thousands of small cells. Really, most of these, all you see is the nucleus of the cell. There's a subcapsular sinus right here, underneath the capsule, between the capsule and the majority of the medullary tissue. The lymphatics coming to this gland approach the periphery of the capsule and enter on the periphery. Then they filter through the gland, the lymph fluid itself, and exit at the hilum. Now notice how this lymphatic tissue is arranged. It's arranged in centers. This is called a germinal center, and there's less cells in the immediate area here in the center, and then there's more cells on the periphery. Let's scan around and see if we can see some more germinal centers. There's one right here. You see how it's arranged? Now, let's go towards the hilum. There's connective tissue, cords that bring in the vessels and the veins into this area. And you see here's some major vessels entering and exiting. And it's the hilum area where the efferent lymphatics exit from the gland. So I think that's enough for the lymph node. Remember, it has a capsule, and it's arranged with medullary tissue here, and then on the periphery we have the capsule. Now the next tissue I want to cover is lymph nodules, or diffuse lymphatic tissue, and that's the same kind of arrangement of the cells, but there's no capsule. So we'll look at that next. throughout the GI tract, there's many times aggregations of lymphatic tissue. Now in the lamina propria here, we see a lot of blue stained cells. These are individual lymphocytes migrating through the lamina propria and then through the epithelium. This happens to be a section of ileum. All along the digestive tract we're going to find lymphatic tissue. Now here, you see in the lamina propria, here's a large aggregation of lymphatic tissue. Now this is a germinal center here to the left. There's no real capsule around here. This happens to be epithelium that's limiting it on the right, and then on the left is, of course, muscularis mucosa. Notice the thousands and thousands of cells found in here. Now in this particular section of the, or portion of the GI tract, the ileum, when we have this diffuse aggregation of lymphatic tissue, we call this Peyer's patches. We'll find this many places throughout the body. This is a lymph nodule. No capsule, no organization of the connective tissue carrying blood vessels in and out of this area. But you do have germinal centers. Here's one in the center of the field now, and there happens to be another one right here. Okay, that's probably enough to distinguish that particular part of the lymphatic system. Now we'll go to the spleen. The spleen is unique in that it doesn't have lymphatics, but it has lymphatic tissue in it. Notice here's a couple germinal centers in this field. It's really a huge reservoir for blood. And we have two areas in the spleen. We have the lymphatic area and we have the sinusoidal area. Now in real life specimens, the aggregations here of this lymphatic tissue appear white, and that's called the white pulp. And the other areas that surround the rest of the field are nothing more than vascular sinusoids full of blood, and of course that appears red, and that's called red pulp. Now notice a good capsule, good thick capsule on the spleen. There's a lot of elastic tissue in this capsule. In areas of hemorrhage or when the body needs more blood, the spleen actually can contract and it serves as a reservoir for blood for the rest of the body. Here's some more of the white pulp, and most of this is red pulp. Now, one thing unique about the areas of the lymphatic tissue in here, we'll always find an artery associated with that. It's called a central artery. Many times it's a little eccentric from the center. I want to show you one of those at higher power. This is a area of white pulp. Notice the artery right in the little off to the center of this area. These germinal centers perform the same function as the germinal centers that we found in the lymph nodes and the lymph nodules. Now in the periphery here we have the red pulp, and you can see these vascular channels full of blood, giving an appearance, red appearance. I want to show you a higher power of these sinusoidal areas in the red pulp of the spleen. We have blood in the sinusoids, and notice the character of the sinusoidal lining cells. Many of these are phagocytic. They are a little larger than usual endothelial cells. Let's look around. This is some that's somewhat longitudinal cut, and I think we can see, here we are, several here that are cross-cut splenic sinusoids. Now in the areas surrounding these we have many phagocytic cells. Perhaps you can see the granular nature of some of these macrophages, phagocytic cells. This is one way that the blood has of conserving iron. is for the worn out red blood cells somehow are sensed and are phagocytized by the spleen, and it's one way the body has of conserving iron. So the spleen has many functions besides just the storage of blood. These are cross-cut splenic sinusoids in the red pulp. Now here you can see many of the macrophages in this particular section. I think we have one more power. It'd be interesting to go up to the next power and see these particular cells. There are several cross-cut sinusoids, actually 4 prominent ones in this field. Notice the lining cells, and then in the center we have 4 or 5 prominent macrophages. You see the engulfed material. Here, here, and here, and here. All over. This is the red pulp of the spleen. Vascular area, the sinusoid to the right, and then there's connective tissue and a stroma of macrophages and other cells found on the periphery. Another cross-cut splenic sinusoid. Okay, now we'll go to another section of spleen just to reconfirm the white pulp areas. We've seen the histology and morphology of the red pulp. I'd like to re-emphasize the areas of the white pulp now. The white pulp follows connective tissue trabeculi carrying in major blood vessels, but in the peripheral areas we have the blood vascular system breaking down into arterioles as it is here, and this is what's called a central artery. This of course is a high power, and all these cells out here are part of the lymphatic series of cells. Small lymphocytes, macrophages, reticular cells. Form the same kind of a cellular function gland. They produce cells actually, as we find in the lymph nodes and the lymph nodules. This is part of the white pulp of the spleen. Thousands and thousands of small cells, strong nuclei appearing in little cytoplasm, in addition to the regular reticular cells, macrophages, and plasma cells. White pulp of the spleen. Now I want to show you some distinguishing characteristics of the thymus. The thymus has a outer cortex and an inner medulla, and the medulla invaginates the cortical areas. Notice the lack of a real strong capsule on the outside here, which distinguishes this immediately from the spleen and the lymph node. Now the light-stained medulla, see now here in this particular section, invaginates the outer cortical areas. So then in cross-cut sections you'd get sections that would appear such as this, with the medulla in the middle and the cortex on the outside. It's almost like you'd section say a mulberry or strawberry. You'd get this kind of an appearance. Little capsule, good areas of many, many cells in the periphery. That gives you the dark basophilic appearance, and then a less staining appearance of the medullary area. Now we'll look at this at higher power. There's a thin capsule on the right, the cortex with thousands and thousands of thymocytes. That's the predominant cell in the thymus. They look just like lymphocytes, small lymphocytes. And here we have the medullary tissue. This is medulla here, and then on the periphery, the cortex. I want to show you some other distinguishing features of the thymus, particularly these hyaline-like bodies that we see right here in the center of the field. These are called thymic corpuscles or Hassel's corpuscles. In order to get a good detail on those, we'll go to the next power. Here are 4 thymic corpuscles in the center of the field. There's some artifact here in this one here below where the knife blade cutting the section has moved these hardened objects to the side. But notice they're concentric in shape. They're formed of dead cells in the thymus. These, of course, increase with age. There's several. There's a blood vessel to the left, and immediately to the right of it is a concentric appearing thymic corpuscle. And here's several in here. Function of these really is not known. See how they're circular in appearance? This is a unique feature to the thymus. Once you see these, you know you're in the thymus. Here we are, again, seeing the huge numbers of thymic cells. These are thymocytes found in the thymus. This is out now, of course, in the cortex. I want to show you what happens to the thymus with age, because there's some retrogressive changes that take place. The thymus gains more fat and loses the parenchymal tissues, as we're looking at now. I'll show you an involuted thymus of somebody approximately 40 years old in the next slide. This is an involuted thymus. Notice all the fat adipose tissue. It's infiltrated the parenchymal tissue of the thymus. The loss of all the thymocytes and really the integrity of the medullary cortex distinction. There's big thymic corpuscles here. We'll scan around. And very little parenchymal tissue left. Notice the large amounts of fatty tissue. Now that's what an involuted thymus looks like. I'd like to review for you the lymphatic tissues. One distinguishing characteristic is the large numbers, the heavy aggregation of cells that we have in this lymphatic tissue. In most of these cells, the nucleus is the largest component. You have very little cytoplasm. So we have very basophilic appearing areas. In fact, in many situations, you can look at the tissue grossly with the naked eye and see the lymphatic tissue. There's several distinguishing characteristics of each of these, and I'd like to review those for you. Now, the lymph node has a good capsule on the outside. It's in the outer peripheral areas that the lymphatics come to the lymph node. Then there's a hilum area where the blood vessels enter and exit the lymph node, and the exiting lymphatics exit, the efferent lymphatics. There are large numbers of germinal centers, and this is a prominent situation found in the lymph node. Now, if we take a lymph node and delete the organization of the hilum and the capsule and the trabecula that course through the center of the lymph node, we have diffuse lymphatic tissue or lymph nodules. The spleen has a very thick capsule. It actually doesn't have lymphatics itself, but it is composed of lymphatic tissue, and this is the white pulp. In the peripheral areas of the white pulp, we find germinal centers, and one of the characteristics of these germinal centers, we find a central artery there. In the remainder of the spleen is the red pulp. We showed you the sinusoids in the red pulp, and the stroma, the macrophages and fibroblasts and other connective tissue cells that we find in the stroma. The cells lining these sinusoids, many of them are macrophages themselves. The thymus is another lymphatic tissue. It's unique in that it has a small capsule, very thin capsule, and an anastomosing medulla that invaginates the cortical areas and gives it a unique appearance. Another unique appearance of the thymus, of course, is the thymic corpuscles, various eosinophilic, concentrically appearing hastles or thymic corpuscles. One lymphatic tissue that we did not cover, of course, was the tonsils. This concludes our discussion of lymphatic tissue. I would like to review for you the microscopic appearance of the digestive tract from the pharynx to the colon. We'll pay particular emphasis to the epithelium that lines the digestive tract. We'll take it from the esophagus, high esophagus, low esophagus, stomach, small intestine, and colon. Now I want to show you a low-power section of high esophagus. High esophagus has skeletal muscle in the muscular portion of this tube. And the epithelium is lined by stratified squamous epithelium. Here we are, epithelium to the left. There's also some mucus glands, as we see in the middle of the field here. You can see the typical epithelium, stratified squamous epithelium. It's moist, it's not keratinized. Now down in here in the muscular area is skeletal muscle. Now here's a good section of skeletal muscle cut in cross-section and longitudinal. Now up to the next power. This is a high-power section of the epithelium of the esophagus. You see the flattened cells on the surface. This is a stratified squamous epithelium. And let's come down in here. Here's our typical mucus glands with the flattened nuclei against the basement membrane. Here's some lymphocytes, a few localized aggregation of lymphocytes. Find that typical throughout the whole digestive tract. And of course this is skeletal muscle. See the nuclei on the periphery of these groups, these individual fibers. This is cross-cut skeletal muscle. Here's even a better depiction of it here. And then this, of course, is some longitudinal skeletal muscle. Wonder if you can see the cross striations at this power, or the really longitudinal striations. Well, that's the upper esophagus. Now I'll show you a section of the esophagus as we go further down the digestive tract. It becomes an increased amount of smooth muscle replacing the skeletal muscle. This is the same power as we had before, showing you lower esophagus. This is the epithelium, a stratified squamous moist, non-keratinized type of epithelium. And let's see what kind of muscle we have here. That's smooth muscle. Longitudinally cut. And here's some. Now we're getting more of an organization of the bundles of the muscles. So we're going to eventually, as we come into the lower areas of the digestive tract, have a real organization of the muscle layers into a inner circular and outer longitudinal area. I want to show you a higher power of this smooth muscle. It looks like it's pretty well organized here. This is longitudinally cut smooth muscle. All you see is a spindle-shaped nuclei. The outline of the cytoplasmic membrane is very indistinct. And I think over here, yeah, here's some cross-cut smooth muscle. Just the nuclei. That's all it appears. Little connective tissue stroma between these groups. And here's something that's kind of unique. This happens to be a sympathetic nerve plexus. And we're going to find these, it's called a myenteric plexus, between the layers of smooth muscle throughout the gut. These large cells are actually autonomic nerve cells. It's found interspersed between the inner circular layer and the outer longitudinal. We'll see that throughout the GI tract. Now on this same slide, we'll go back to the epithelium and actually return to a smaller, lower power actually. Here's the epithelium. And I want to trace this on to the cardioesophageal junction. There's the free border. This is the esophagus again, stratified squamous epithelium. Notice here's one of these diffuse areas of lymphatic tissue, a lymphatic nodule. All right, now tracing this epithelium, all of a sudden, still stratified squamous, there's some more lymphatic tissue. We have an abrupt change of the epithelium from stratified squamous to one that's a columnar epithelium, and this is typical of the cardiac region of the stomach. This happens to be all tall columnar mucus cells. A lot of lymphocytes in the lamina propria. Okay, I think it's probably appropriate that we change power, or at least look for a more typical area of a gastric crypt. This is the cardiac portion of the stomach, and these are mucus cells. They're on the epithelium, and actually even down in here where we have the branching of these crypts, we've got mucus cells down in here. Now this is the so-called gastric pit, then there's a neck. Because this is a branched tuboalveolar gland, we have a lot of cross-cut sections down in here. This is all epithelium, and then there's a core of connective tissue, of course. This is a typical epithelium found in the cardiac region of the stomach. See, the pit is only about a fourth of the way down into that gland. These are glands of the stomach, but they're just epithelium lining the surface of this tissue. Now I want to show you the main body of the stomach, the epithelium that lines that. This is in the fundus or body of the stomach. It's really necessary to scan these sections quite a bit to find a typical area of the epithelium so that you can see the outline and description of the pits. These are the invaginations of the epithelium. Then there's a neck area. And at this power, I think you can see right in here all the cross-cut sections that you have of the branching of this particular gland. I want to go to another power now. We've moved to another section. The lumen is on the right. That's mucin that's on the top of the cells, the epithelium here. This shows the tall columnar cells of mucus cells that are in the pit of this stomach. Now about halfway down we have the neck of the stomach, and then you see all the cross-cut sections here of this branching gland. Now I want to show at a higher detail for you the different kinds of cells that we have in this gland. This is the lumen over here to the right, and this shows you the typical tall mucus-secreting cells of the stomach. Then there's the neck area where the cells become shorter. They're called mucus neck cells. And then down when the gland starts to branch, we're going to have several kinds of cells. Some are going to have granular cytoplasm, as we see these cells in the center of the field. See the granules. Now that's the chief cell. And then there's going to be other rounded cells that are big. There's some rounded cells there. Those are the parietal cells. So the ones with the granules are the chief cells, and then the ones that are big and round. Here's one here, and here. Let's see if we can find... Oh, here's several. Right in the center. You see the granules in the cytoplasm of these chief cells. Then there's other cells that have intracellular canaliculi, and they're called the parietal cells. The chief cells secrete these zymogen granules, and they contain preformed enzymes. The parietal cells secrete hydrochloric acid. And here's a bunch of chief cells. See the eosinophilic zymogen granules? Let's see if we can find some more good parietal cells. Big, large, pyramidal-shaped cells are the parietal cells. We've shown you the typical cells found in the main part of the stomach. These are the surface columnar mucus cells, and there's smaller cells that are the mucous neck cells, and then the two other prominent cells found in the branching crypts of the stomach. And this is the parietal cell that secretes hydrochloric acid, a big round pyramidal cell, lightly stained, and the chief cell, which secretes zymogen granules, and you see the characteristic eosinophilic cytoplasm, apical cytoplasm of these cells. There's another cell found in the stomach that stains very well with silver, and that's the arginophin cell. And we didn't show you that, of course. Now I'd like to show you the area of the stomach called the pyloric region. It has a unique type of epithelium. This is the epithelium of the pyloric region. Now the pits are real long here, almost halfway down the epithelium. And then the epithelial cells themselves are all mucin cells. So this distinguishes it, of course, from the main part of the stomach. But the depth of the pit is a characteristic, an important characteristic to distinguish this pyloric region of the stomach. Very briefly, I want to show you the high power of this epithelium. Lumens to the left, and you see the surface epithelium. These are tall columnar cells, the lumens coursing right through the middle of the field. See the lamina propria in here. It's a good place to find plasma cells. Let's trace this long pit, you see, down to here. Now this would be the neck of the gland. You can see how it's branching here. We come down into the crypt area, and you see we still have mucus cells, even down as far as the muscularis mucosa, which is right here. See that smooth muscle? So this is a typical epithelium of the pyloric region of the stomach. Mostly mucin cells. There we are back out to the surface, or the free lumen area. Next thing I want to show you is the duodenum. There's a large bundle of smooth muscle in the pyloric region of the stomach, in muscularis area, and that's the so-called pyloric sphincter, but it's made up of smooth muscle. No need to show that here. Next is the duodenum. The epithelium of the duodenum is in the middle of the field. There's debris and mucin on the right, then the epithelium, then there's muscularis mucosa, and then you see there's some submucosal glands here. And these are mucus glands. Muscularis is over here, muscular coat. This is submucosa, muscularis mucosa, and then the epithelium. These are mucus glands, and they're typical of the first part of the small intestine. They're called Brunner's glands, or duodenal submucosal glands. I want to take a closer look at the epithelium now of this first part of the small intestine. The epithelium in the first part of the small intestine is the same as found in the second and third parts, the jejunum and the ileum. Just for clarity, I've switched to a section of jejunum to show you the orientation of the villi. That's this area on the luminal side, and then the crypt area. Notice that you don't have the number of cross sections of the crypt area as you do in the stomach. It only branches a few times, the villus and the crypt. This is jejunum. Now the goblet cells are going to increase in number. The surface epithelium is a tall columnar epithelium, and I want to show you a high power of that. This is a villus in the jejunum, has a connective tissue core that carries the vessels in and out of this area. You know that this is an area of high transport of nutrients that pass through the tall columnar cells into the connective tissue area. There's also a blind lymphatic, the so-called lacteal, in that connective tissue. Notice that the cells are tall. It's a simple columnar epithelium, and there's a distinct striated border on the apex. That's caused by microvilli at even height, tall columnar cells. They come down in here. Here's the branching into the crypts, and then you have very few cross-cut sections down in this area. To the left of the field, of course, is muscularis mucosa. Several goblet cells are found here. There's also a unique cell found in the crypt area, basement area of the crypts. That'll be the Paneth cell. That you can tell from the bright eosinophilic granules in the apical cytoplasm. So this is what typical epithelium of the small intestine is like. It's a simple columnar epithelium, and there are numerous goblet cells found throughout. Now in the ileum, which we are showing here, same kind of surface epithelium. See the core of this villi, or villus, and as we proceed down the digestive tract from the duodenum to the colon, we have an increase in the number of goblet cells. These are these faint staining apical cytoplasm that you see, the rounded areas. We still have the tall columnar cells. This is ileum. Now I want to show you a section of colon. Colon is the name given to the large intestine. The surface epithelium has no villi. It's just one straight crypt. And perhaps at this power you can see the high concentration of mucin goblet cells. This is a higher power of a base of a crypt of Lieberkuhn, of an intestinal crypt. Tall columnar cells, and you'll notice that there is a high concentration of the mucinous goblet cell. Now in review, we've stressed primarily the surface epithelium of the digestive tube because it's unique to each of the areas. We saw in the esophagus, it was stratified squamous, moist epithelium. There were skeletal muscle in the upper third, merging into a muscular coat of smooth muscle in the lower third of the esophagus. The stomach, we showed you three parts: the cardiac region, the fundic or main region of the stomach, and the pyloric region. It's unique in that it has pits, and then there's a branch tuboalveolar gland, so that in the crypt area there's many cross sections of the gland itself, of the surface epithelium. The duodenum is unique in that it has a simple columnar epithelium, tall columnar cells with a striped cuticle or striated border. In the submucosa, we find mucus glands. These are Werner's glands. Going on through the small intestine, we still retain the simple columnar morphology of the epithelium. However, the goblet cells do increase. The epithelium is arranged so that there is a villus and crypts, but the crypts are not as cross-cut. There's not as many seen in cross-cut section as in the stomach. Now in the colon, we have a simple crypt, a test tube-like crypt of Lieberkühn, and we have an increased number of goblet cells in the epithelium. That completes our review of the digestive system.
You've been listening to a presentation from the University of Michigan School of Dentistry, which is dedicated to supporting open learning and open educational resources. This recording is licensed under the Creative Commons. It may be reused and redistributed for nonprofit use. Please attribute materials to the University of Michigan School of Dentistry and redistribute under this same license. For more information on how this and other University of Michigan School of Dentistry recordings may be used, visit www.dent.umich.edu/license.